Hello everyone, it's me Mole, and today this is me going over the study guide for Unit 1 for Biology. Alright, first things first, if you go to the actual assignment that she provided, she said, note for Bio Chapter 1 worksheet, follow page 48, Quarter 1, page 49 to 51, disregard the rest of the questions on the previous pages. So we're going to skip through all of these because she said to focus on that. If you do want to know them, let me know. I'll teach them, but let's just focus on what she provided. So she said go to page 48, which I believe is... hold on, let me open it up. 48 is this one. I skipped it, my bad. Alright, yeah, this is 48. Alright. Match the following terms with their correct description: endergonic reaction, exergonic reaction, metabolism, and metabolic pathway. The sum of all the chemical reactions in an organism, let's just skip that for one. Okay, the reaction that gives off energy, right? That gives off energy. So what is an action that gives off energy? So an action that gives off the energy, or aka releases energy, okay, is an exergonic reaction, right? So if we ever look at a diagram of an exergonic reaction, okay, it looks something like this, all right? What this means is this is the starting reactants. This is the energy that it requires to actually create the chemical reaction. This is like the activation energy per se-ish, right? And then these are the products, right? So you can see how energy, so this is energy. Energy is all... oh, actually, no. Energy, that's the wrong area where we put energy. My bad. Energy should be on this side. So as we see, energy starts to stay here in the middle, then goes really high, but then drops. So that dropping is energy being released. So energy being released, a reaction that gives off energy, is exergonic. Alright, so always remember that. So that kind of like lets you know what the next one is, and a reaction that requires an input of energy. That's endergonic. Okay, so an endergonic reaction looks something like this. Oh, I'm sorry about that. So an endergonic reaction looks something like this, right? Where energy must be absorbed. So this part is energy. It must be absorbed to actually do the chemical reaction. Now let's see. Let's now see what the next one is. Metabolic pathway. Right? What's a metabolic pathway? So metabolic pathway is basically a series of linked series of chemical reactions occurring within the cell. You can say that it's the interconnected chemical reactions that occur within the cell. Almost all the reactions that's going to happen, right? A series, right? So metabolic pathway, what is a pathway, right? A pathway is a series of steps, right? So a series of reactions that either break down or build a molecule would be a metabolic pathway, because it's a path to making these events happen. So this answer would be metabolic pathway. All right. When it comes to the sum of all chemical reactions in an organism, that would be metabolism. Metabolism requires both the intake of energy and the releasing of energy. All right. It's not a series of steps. It's the general whole thing, right? So that would be metabolism. So that's the question for number 5. All right. A chemical reaction is determined to be exergonic. If you're trying to get that reaction to occur using additional and a separate reaction, a chemical reaction is determined to be endergonic. If you were trying to get that reaction to occur using an additional and separate reaction, what would you do? All right, so let's again go back to what endergonic means, right? So let's first make the proper diagram so you guys can get a clear idea. All right, that was wrong. So we're gonna make a XYZ graph. This will be free energy. Or energy, actually let's just call it energy. We're not going to go over Gibbs because that's a bit too advanced for us. So that'll be energy, and this part would be time. So an endergonic reaction goes something like this. Alright, actually, you know what, that kind of looks bad. Looks something like this. Alright, so this point would be the reactants. Alright, so that would be the reactants. This point would be the products. This area would be energy required to actually make the reaction happen, right? This here is more specifically the activation energy, but this area in general is the energy being needed. Again, this requires energy to be inputted to allow us to do the reaction, right? So the question is asking, a chemical reaction is determined to be anorganic. If you're trying to get that reaction to occur using an additional or separate reaction, what would you do? So what would we do? Let's say there's a reaction that's exergonic here, okay? So an exergonic reaction. That means it's going to release energy. So what does that mean? An exergonic means releasing energy. We would take that released energy. Oh my god, I'm so sorry. All right, we would take that released energy, released energy, and used it in an endergonic reaction. Because again, endergonic needs energy to occur. Needs energy to occur. You can always think that exergonic and endergonic are hand in hand. Exergonic produces the energy to be used in endergonic reactions. They're always going to be hand in hand. So if we're going to say like a chemical reaction is determined to be endergonic, if you're trying to get that reaction to occur using an additional and separate reaction, what would we do? We would take an exergonic reaction and take the energy that is released to be used in an exergonic reaction and create the products. That's how it happens. Alright, that's how number 6 has occurred. Let's do number 7 now. The phosphorylation, this is a very key word. Phosphorylation. I want you all to know that. Why the hell is it? Okay. Phosphorylation of ADP creates blank. What does phosphorylation mean? It means a phosphate is added. A phosphate is added. So let's look at the diagram of ATP all over again. ADP actually, because we're looking at ADP. ADP stands for adenine diphosphate. So it would look something like this. So let's say this is the adenine group. This is one phosphate, and di means 2, so there's only 2 phosphates. This is a low energy system, meaning this won't provide energy to do chemical reaction. But the phosphorylation or the adding of ATP would make it to adenine triphosphate, aka ATP. Now this ATP can be used for chemical reaction because it's energetically unstable, so they need to release the energy. So we would say the phosphorylation of ATP creates ADP. Remember, ADP to ATP through phosphorylation. And then through hydrolysis, it's ADP. A cellular pump creates certain items against their concentration gradient to maintain those gradients. What type of cellular work is this considered? All right. So a cell pump contains certain ions that are against their concentration gradient to maintain those gradients. What type of cellular work is this considered? So let's understand this. What is the concentration gradient? What is all of this meaning, right? So let me break it down for you real quick. Alright, so let's understand how it works. So this is what we should have learned in Suarez's class, but if you didn't understand, let me explain it to you right now. Alright, so let's look at a phospholipid bilayer. I'm sorry if my Apple Pencil is like looking like that. It's just an old rusty one. Alright, this is what our phospholipid bilayer sort of looks like. Alright, and if we should already understand how a phospholipid bilayer should look like, right? Phospholipid bilayer is very specific in the sense of no polar molecules or water-liking molecules can get through. As we already know, the tails are hydrophobic, meaning they hate water. And since they hate water, this basically means that items that like water, such as salt and sugar, can't get through the phospholipid bilayer. So this means we need proteins to help us get it through. So these proteins are embedded through the entire phospholipid, allowing these molecules to get through. But now let's understand concentration gradient. Now, I'm going to simplify this diagram a little bit more so I can like draw it better. Let's say this green line are the phospholipid line. Whatever this thing is, it's just this green line that we're representing. This red shows a protein. Now, the way it works is pretty simple. Let's say that we, to describe what osmosis is or the diffusion is, let's say that we had a jar of water for a second. Bear with me for a sec. Let's say we had a jar of water, okay? This jar of water was just filled with normal water. Now, let's say we added a drop of red food color inside the water. What would happen? What usually happens isn't that the water all of a sudden turns red. Instead, the water turns murky red, almost diffusing with the water. And then if you mix it, it does turn red, right? So it's the same thing happening here. what usually happens with the cell membrane is that there is a bunch of these molecules that want to get through the phospholipid bilayer. So these are a bunch of molecules that want to get to the other side. Remember, this is the cell membrane. The green is the cell membrane. So because of diffusion, it is easier for them to go from something of a high concentration to a low concentration. To put in layman terms, if there's a high concentration of something, so if there's a high concentration of molecules, there's a high concentration of, I don't know, balls or anything, it requires no energy to go from high concentration to low concentration. So high concentration to low concentration requires no input of energy because we're just going with the flow. It's just how the cookie crumbles. All right. When we put the drop of water into drop of food coloring into the water all of a sudden doesn't just magically turn red. It sort of diffuses and turns red after a while. And we're not putting any energy into it. It just happens on its own. But let's say this is the complete opposite. Let's say instead of it trying to go through and requiring an input of energy, let's say it's a high concentration and more want to go on the other side. Well, energy is going to be required. All right, energy is going to be needed to push the molecules from an area of low concentration to high concentration. So listen to me very carefully. So if we're going from a high concentration of molecules to a low concentration of molecules, there is no input of energy. But if we're going from a low concentration of molecules to a high concentration of molecules, an input of energy must be required. And since we're doing that here, we're going from against their concentration gradient. Against their concentration gradient means high to low. We require an input of energy. And this input of energy is called active transport. All right. Miss J. Patel will go way more into detail with this, but this is active transport. If you want to go a little bit more nitty gritty with the actual concept, it is using the type of cellular work that's considered is a usage of ATP. But the actual answer should be active transport. She should be given credit for this. Again, you can ask her, talk to her about it. All right, let's go to number 9, the multiple choice, finally. Anything that inhibits ATP synthesis in a cell would most likely force the cell to use ADP as an energy source. That can't be the answer. As you already know, ADP is too energetically low to provide any work. Result in the death of the cell. That could be possible. Make the cell start using lipid as energy. Maybe. Cause the cell to stop doing mechanical work, but not transport work. Now, I would say D should be a solid answer. I'm right now going with A or D, a B or a D. Now, ATP is a very critical part of the actual exam, but you have to understand that. Wait, I think it's B, guys. Let me explain why. So the reason why I think it's going to be B as your answer choice for this problem is quite simple. It will cause the cell to stop doing mechanical work. That's a given. No matter any work that we do, such as when we start doing cellular respiration, ATP is required. Photosynthesis, ATP is so much chemical reaction required. But not transfer work. But not is a pretty important thing. Because as we just explained, active transport requires ATP to happen. So if these transport work requires ATP, don't you think the transport work would also stop working or be in a very dormant level? And because of that, the answer has to be B. It will result in the death of the cell. That's why ATP is so critical. It's such an important molecule. If it's not there, we are dead. Okay? So that's number 9. All right. Answer the following as you read modules, yada, 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 to yada, yada, yada. All right. Energy needed to initiate a chemical reaction. You guys should know this. This is activation energy. If you look to a diagram of anything requiring activation energy, let me just draw it again. It looks something like this. This portion is our activation energy. All right. I'm sorry my thing is so bad. It's just, my pencil. I'm sorry. All right. Let's go to the next question. This was a pretty simple one. In pole vaulting, a higher bar is placed and it is more difficult to clear. Explain why this is a good analogy to help students understand enzymes and activation energy. Wait, we did this question in class, so this means you should be pretty well set in answering this question. I do believe she gave this as a do now for once. I don't remember clearly, but let me see if I can find that do now, because I'm kind of lazy to write my answer. Is it worksheet 1? I think so. No, it isn't. My bad. Alright. Okay, I'm just gonna write down the answer. Okay. In pole vaulting, the higher bar of space, the more difficult it is to clear. Explain why this is a good analogy to help understand enzymes and activation energy. Let's look at a normal chemical reaction. Let me draw an example. It would look something like this. Alright? And then this is the one with activation energy. This represents the time needed to do a chemical reaction. Alright? Or actually I drew the diagram wrong. Give me a second, I'm so sorry. Alright, let me redraw that. So this would look like, this would be the example. This would be the example of a chemical reaction done with activation energy, and this would be the example of a chemical reaction done without activation energy. First of all, this is a time plane, by the way. As you can already tell by this graph, it requires more time to already do this reaction. So that's already going to tell you why it's harder. Not only that, it requires more energy for the reaction to occur. Activation energy allows us to almost kickstart the reaction, letting us require less energy overall to do the reaction. So when it comes to this question, in pole vault, the higher bar is placed, the more difficult it is to clear it. Enzymes lower the activation energy, allowing less energy required to actually do the whole chemical reaction. This will make it easier for the chemical reaction to occur. That's why it's a great example, right? So enzymes lower activation energy, aka it's almost lowering that pole bar, which makes it easier to clear through, which makes it easier and faster to occur to the chemical reaction. That's usually how I would frame this question. I think that's the best way to understand it. If you need any help, again, ask me. Alright, graph on page 87 in your textbook illustrates the difference between a chemical reaction with and without an enzyme. Could the reaction represented by the black line still occur without the enzyme? Explain her answer. First of all, we already did this question already, but let me just go through it. The answer is yes. Yes, it can occur. But how so? The best way to know that if a reaction occurs is look at the product stage. If the product occurs, if this line reaches the product line, this white line right here, let me zoom in. If the line of this graph reaches a white line, that means products are going to be made no matter what. Sure, it will require more energy for that reaction to occur, but it's going to happen. So that's why, yes, it will always happen. You have to understand chemical reactions will always occur. They either need a kickstart of energy or need a really, really long time. All enzymes do is just speed up the reaction. This doesn't mean that all of a sudden the products are going to be made or not. It just makes it easier for it to occur. Let's do the next question. Enzyme, active site, and substrate. I think we already did this question. It should be key and lock, right? If we look at an enzyme, let me draw a quick enzyme right here. Let me draw a quick substrate right here. It's almost like a key and lock fitting in for the actual thing to occur. Alright, circle the enzyme. In the following reaction, circle the enzyme. Here's the fastest trick to ever finding an enzyme. If it ends with ASE, it's an enzyme. So it's going to be lactase. Always find ASE to determine if it's an enzyme or not. Alright, let's do the next one. Alright. Damn it. Can I move this damn thing? What the heck? Alright, my bad. Let's do number 6. Okay. Sucrose is an enzyme that breaks down. Sucrase is an enzyme that breaks down sucrose to glucose and fructose. Will sucrose catalyze the breakdown of disaccharide maltose? No. Why would it not? Okay. So the answer is very simple. Enzymes are specific. That means an enzyme is only connected to their function, right? If sucrase can only break down sucrose, it will only break down sucrose. So if you're thinking, oh, it might break down maltose, no, there will be a separate enzyme for that. Enzymes are highly specific. They're very specified. So that means it needs to, it can only work for a specific function. So no, it will not help catalyze a breakdown for the disaccharide maltose. Alright, create a Venn diagram to comparing inhibitors. Alright, I'm not going to create a Venn diagram. Instead, I'm going to explain how this works, okay? Let me explain how this works. Alright, let's say we have two enzymes. We'll call them enzyme A and enzyme B. All right. So let me draw the enzymes real quick. This is going to be the enzyme. So what does competitive inhibitors do? So let's say this is our substrate right here. So this substrate is going to try to go here. This is their active site, right? Active site is a place where the substrate gets attached to on the enzyme. So what happens is the end substrate should usually go to the active site and boom, the product will be made. Competitive inhibitors, they come in and they stop it. They come into the active site and take over the substrate, blocking the substrate from happening, stopping the enzyme production. That's how competitive inhibitors work. Non-competitive inhibitors are a little bit different. Instead of blocking the active site is still clearly open, but instead, a non-competitive inhibitor will come to another part of the enzyme. This will morph the shape of the enzyme, almost like a activation key, changing the shape of the enzyme to something in which the substrate cannot attach to, thus blocking the production of the enzyme. The similarities between these two is a block substrate enzyme activity, and then from this diagram itself you can see the differences. Alright, let's use figure yada yada. Use figure 515b on page 89 of your textbook to answer the question. Assume that a toxin is introduced to a cell that binds and removes substrate C from a pool of molecules participating in a reaction. Briefly explain what happened to the amount of substrate B. Alright. Assume that a toxin is introduced to a cell that binds to and removes substance C from the pool of molecules participating in the reaction. So it will remove this. Alright? Briefly explain what happened to substance B. They're intermediate molecules, meaning they rely off each other. So intermediate means they're in the middle of the reaction, right? So A turns into B, B turns into C, and then turns into D. What would happen to the amount of substance B? The amount of substance B would actually be gone. Alright, so what would happen is the enzyme is not affected based on what the description is providing. I don't have too much background knowledge to say my opinion, but I think enzyme 2 is not affected at all. So what would happen is B would come in and do the reaction, and then C would be made, but it would remove C. So it would either inhibit enzyme 2 from actually creating, and there'll be a mass amount of B and the reaction won't occur, or B will be made into C, but the second C is made in the enzyme, the toxin will remove it. So either will be a mass amount of B or no B. I really can't explain too much of what will happen, since I don't know if the toxin is continuously removing C, or it removes C and it's a done deal over. I will talk with Ms. Jibatel on this question, and I will come to you to see what I can do to answer it. Let's do the next question. Penicillin is a drug that affects bacterial cells and not human cells. How is this possible? Alright, this is a really key question. Penicillin, how does penicillin work? Okay, penicillin works in a multitude of ways. But how does penicillin work? Alright, so penicillin works by bursting the cell wall of bacteria. So if you don't know, human cells don't have cell walls. Plant cells have cell walls. Bacteria has cell wall, and fungi cells have wall. But each of these cells that have cell walls are each unique. Plant cells has a cell wall made of cellulose, I believe. Fungi have one made of chitin, and bacteria has one made of this weird thing that I don't know what's it called. But the way it works is penicillin just stops the production, just destroys the cell wall. Destroying the cell wall allows it to affect bacterial cells, but not human cells, because human cells don't have walls. They burst cell wall of bacteria. Alright, connecting big ideas. I think that's the one. Wait, let me go back to the unit test. Alright, we go through page 49 to 51, I believe. So I think this is our last page for this topic. Again, if she asks us to do any other question, like in this thing, I will definitely answer them. I just don't know if this is what she meant by it, because I really don't know. But I'm gonna finish it up now. Okay, this says here, aquaporins are membrane proteins that span the entire membrane, transmembrane protein. An aquaporin protein must be able to interact with the polar regions that are both externally and internally, as well as the polar molecule that move through. Additionally, it must be able to interact with non-polar. How is it possible that this protein can meet all these requirements? Okay, this is a complex answer, but I think we can answer it beautifully. All right, first we need to understand what proteins are made of. Proteins are made of amino acids, and amino acids are actually extremely unique. Amino acids have something called an R group. So if we look at an amino acid, unlike carbohydrates and nucleotides and everything else, we see them look in uniformity, right? A carbohydrate, it's just 6 carbons, 12 hydrogen, yada, yada, yada. Same with all of them. But when it comes to actually amino acids, there's something a little bit special about them that I feel like is very key to understand. Amino acid monomer looks something like this, where it's an R to C to H, connected to a CO and then OH, connected to an N to an H to an H. This is the amino acid setup, right? But what does this setup even mean? This is pretty important. If we were to look at every single amino acid that there is in the human body, this part right here will always be the same. Every amino acid is the exact same in this part, except for this R group, because this R really isn't an element. It's a variable, like X in math. X in math isn't actually something. It just represents something else. So what does this R represent? It represents 20 different molecular arrangements that amino acids could have. These 20 molecular arrangements make amino acids a very diverse group. 20 different types of amino acids can be made. And these 20 types of amino acids, so let me write that down real quick. 20 types of amino acid can then be used and set up in a infinite amount of ways. So amino acid number 1 connect with amino acid number 7, yada 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 yada, to this whole chain of events. Right? There's 20 amino acids. I don't know if Ms. J. Patel touched upon it, but that's how it works, right? There's 20 different types of amino acids, and these 20 amino acids chain up in an infinite ways. An infinite number of varieties, right? And specific types of these amino acid groups, their R groups can provide hydrophilic properties, right? If we were to look at an amino acid setup, let me open that up real quick. Amino acid chart. This is a wonderful example. Let me add that here, right? All right. I don't know why it's so large. Let me lower it massively. Oh crap. I don't know why it's so big. Let me just shorten it a bit. Alright, yeah, this should be enough for you guys. Alright, this is the amino acid setup. As we already know, this is how amino acids are structured, right? So every... crap. Alright, so as you already know, this is how amino acids are structured. So every amino acid that there is, this R group can be replaced by any one of these 20 special cases, right? So for example, this R could be something like this, right? It can be something like this over here. Or this R group can be replaced by glutamic acid, or it can be replaced by lysine. But the actual molecular structure of these amino acids also add into their effect. If we look here, we can see how it says positive here. This is how it says negative here. This is how it says polar with uncharged sides. It says special cases. If we continue downwards, it says hydrophobic side chains, right? Each of these are very much important because the structure of those amino acids, those chained up, each have their own amino acid with their own special property. Some are positively charged, some are negatively charged, some are hydrophobic. And because of that, when they fold into actual proteins, they each some sectors of the actual protein itself are hydrophobic. Some sectors are water-loving. Some sectors are water-hating. So when we look at aquaporins, these proteins that help with water transport, there's regions of them that love water, that that connect with the hydrophobic tails. There's some parts that connect with the hydrophilic heads. There's some parts that connect with the water molecule itself. And the structure of the amino acids based on their structure of the protein based on their amino acids allow this reaction to happen, or allows this connection with the other molecular molecules to happen. Now, I don't know how I could write this down because I'm right now using a pen, but this is really just an idea to explain what's going on. I believe that's everything that we need for this PDF. Again, all she said was follow pages 48, quarter 1, and chapter 59 to 51. I don't know what she meant by quarter 1. Let me go through what she meant by that. I don't know what she meant by quarter 1 at all. I'll ask her. Maybe we can do a few of these problems. I feel like we can do them. The food we eat contains chemical energy. Is it possible for a candy bar to have potential energy for two different reasons at the same time? Technically, yeah, but I don't know why. Actually, yeah, this question is kind of confusing. I'll ask her about that, actually. Energy is never created or destroyed. What happens to the energy that was stored in your gas tank after your lawnmower comes to a complete stop outside of running out of gas? It was converted to heat. Entropy decreased in the universe. The lawnmower gained energy. It's definitely not entropy. Entropy is a very complex topic that I'm not going to go too much into. Energy cannot be lost. It was converted to heat. Well, it technically be lost, but that would be free energy. Yeah, now we're going somewhere that we didn't learn. Second law of, which portions shows the second law of thermodynamics? Second law of thermodynamics is, heat will always flow spontaneously from hotter to colder regions. Entropy will always stay in this universe. It's gonna be this part. This part explains second law of thermodynamics. Okay. Actually, it cannot transfer from a low to higher energy without the combustion of... Yeah, no, it is this one. Most likely. Alright. I think that's everything for this PDF. I think I explained every question pretty well. If you have any questions, again, contact me. I'm more than willing to help. This is everything that we need to learn for this specific PDF. There are a few more. I'm going to make a separate video explaining the Miller-Urey experiment. Actually, should I do that right now? I think I might just do that right now, maybe. The Chapter 5 study guide, I don't know what to do because this is a bunch of topics that we didn't specifically learn and she didn't tell what portions of the actual PDF we need to worry about. I'll ask Ms. J. Patel about that and then I'll make a video separately on that. Let me quickly explain the Miller-Urey experiment because it's actually pretty much probably the easiest one that you're going to take. So the Miller-Urey experiment is one of, if not the easiest, concepts that you're going to learn. Let me show you how it works out. Alright, here we have the beautiful Miller-Urey experiment. Let's understand how it works. Alright, I don't know why it's cut off like that. Why the heck is it cut off? Alright, okay. I don't know how I can yeah, I think that's the best. Alright, so this is the Miller-Urey experiment. Why can't I Alright, so this is the Miller-Urey experiment. I don't know why I can't show you this part, but it's just not working really well. Alright, so the Miller-Urey experiment is a pretty simple experiment in the sense that it is all about how life could technically be made in early Earth. So the way it works is let's first look at this part and we'll slowly move around the entire experiment. So this is I like to call the atmospheric area. So these are electrodes. Electrodes then make sparks over here. This provides energy. This energy is in the form of electrical energy. The energy is very important because it provides help into creating chemical reaction, provides help in creating chemical bonds, it helps break chemical bonds. It really is the energy source, the activation energy, to help set up everything. Following here are 4 gases that are going to be found on early Earth. Hydrogen, methane, hydrogen gas, and I think that's nitrogenous oxide. I don't really know. I don't know what NH3 is. NH3 is ammonia. It is actually ammonia. I'm very sorry about that. But methane, oxygen, hydrogen gas, and ammonia. These are gases that are found in early Earth. Now, these gases are then staying there. This is like the atmospheric area. What happens next is now we're going to go through the sampling probe. This contains water. This is water similar to ancient the oceans, right? Salt wasn't clearly formed yet. I think it was fresh water at this time. I'm not 100% sure, but it's just normal water. The water is then heated up. I can't show you that. Let me try. Yeah, the water is then heated up, right, and went to this heat source. The heat source then makes it into gas, which then makes it condensed, and the water vapor then goes up to here, continues going the water vapor into the gaseous room. Again, as we already explained, the gaseous room contains the atmosphere of early Earth, and the water vapor then chemically reacts with the gases from early Earth, and the spark provides the energy that will allow us for these reactions to occur, making these events happen. From there, it will condense into back to liquid, but now it's not water anymore. Instead, it's this water that has many complex molecules such as amino acids, macromolecules such as lipids, and much more. So basically, this Miller-Urey experiment allows us to understand how complex molecules that allow life to be formed actually formed during early Earth. The usage of learning how gases and sparks allowed us to get an insight of imitating early Earth allowed us to get insight on how these complex molecules formed, because we just didn't exist out of nowhere. Something must have occurred for this to happen, right? So this is how like early complex molecules made, which then led to early basic organisms such as bacteria and amoeba and so on and so forth. This is basically how the Miller-Urey experiment works. If you have any questions, again, contact me. I'm more than willing to help. Talk with Ms. Patel as well. That should be everything for the study guide. It should have everything that we need to know. I feel like that's all we need. If you guys need any more help, by all means, talk with me. For the other one, for the other assignment called Unit 5, Tracing Evolutionary History, Miss J. Patel went over that in class, so I shouldn't be going over it with you guys. Again, if you still need help, message me. Yeah, I think that's everything, guys. Thank you so much for watching. Again, if you have any questions, I don't know if I explained this very well. I think I didn't do too well, but if you need any more help, just, let me know. See ya.